Hi, everyone. We have a number of students who are still joining us today, but we're going to go ahead and get started. Good afternoon. Welcome to the first seminar in the Department of Biological Sciences Fall 2021 seminar series. My name is Dr. Shelley Thomas, and my co-host is Dr. Greg Eaton. We represent two committees who are sponsoring this event, the Seminar Committee and the Student Professional Development Committee. And we're very pleased to have you here today. This session is being recorded, just so you know. So our seminar series includes 4 more seminars this semester after today. You can see here that we have this poster that is hanging up all over the department. So anytime you want to look into more of this, you can click on this QR code. We have 2 research presentations. Dr. Keck from MCB and Chemistry will be presenting his research on developing a drug to address addiction. And Dr. Brian Beatty will be presenting his research on whale skull evolution. And we also have something new this year. We have some career seminars. And one of our goals is to encourage our bio majors to explore the breadth of possible career opportunities open to them. And so we have two presentations on that. So Dr. Helen Paxton and Chris Gravante from the bio department are both specialized medical technicians, and they'll be discussing the role of both general and specialist medical technician scientists. And then Dr. Jan Gorniak is the chief medical examiner in Las Vegas, and she has been involved in a number of famous cases, and she gives talks around the nation. So we're very pleased to have a chance to have her speak here at Rowan. When you check out this poster, you can find that there's a QR code down here at the bottom that will take you directly to the Biological Sciences Department calendar, but you can also get to that calendar directly from the website of the Department of Biological Sciences. If you just scroll down a little bit, you come to this calendar here and you click on it, it'll take you to a calendar of events that gives you all kinds of information about different clubs and different workshops and different seminars and things that are going on in our department and in some of the other science departments. So for example, here's today's event, but I just want to point out that we also have a workshop coming up tomorrow at 7. If you click on that, it'll give you the name of it. It's about career options and job searches. And then you can also click on this link that will take you to the place to register for that event. So I wanted you to know that there's different ways to get the Zoom links to come to these seminars and to see a lot of these other things going on in the department. So in today's seminar, we have a panel of our department's researchers. Please note that not all of our researchers are represented here. And also you can find their specific contact information on the departmental website if you check under faculty. So here's the format of today's seminar. Each presenter, each panelist will briefly present their information about their research, their results, and then especially what students do in their lab. And then after after all of them have presented, we will have about 15 minutes of a Q&A session. So we encourage you to write questions in the chat, and these questions will go directly to myself and Dr. Greg Eaton, and we will ask them during the Q&A. It's best if you write these during the presentation so we have time to organize them for the Q&A, though of course you can also post them during the Q&A. During the Q&A, you can also speak up. However, if you are doing this for participation for a course, you need to type them in and not just speak up. Since this is a panel, when you type your questions, please make sure that you start the question with the name of the panelist to whom you are asking the question. If it's for everyone, just say that. And if you're doing this for participation in a class, make sure that you include the instructor's name in there and then follow any other instructions that your professor gave you. So you can't see anyone else's Q&A or the questions that are being asked, but just know that we are receiving them and organizing them. Okay, so now for our panelists, I am pleased to introduce our first panelist. This is Dr. Stephanie Spielman. Go ahead and share your screen, Stephanie.
I can't share my screen while you are sharing. It won't let me.
Oh, you were supposed to be able to just do that yourself. Thank you. I stopped sharing.
Here's PowerPoint, and hopefully, see if we can get this. Does this, how to, it's, oh, hold on, it hid the rest of my screen. Can you see? I can't see what you see. What do you see?
It was up a second ago, and now we see you again.
Okay.
So it looks like you stopped sharing your screen.
No, I'm still sharing the screen, but the PowerPoint didn't want to appear. So it's a bit tricky to get it into the share mode. So I'm just gonna present like this, if that's okay. Cool. Hi, everybody. Thanks for being here. My name is Stephanie Spielman, Dr. Spielman. I'm a professor in the Department of Biological Sciences. I'll tell you a little bit about what I do and what you could do if you were to get involved with my lab going forward. So what I do, my research focuses on a field called computational molecular evolution. In other words, computational, I use the computer. I don't have a wet lab. I don't use Petri dishes. I don't use pipettes. I don't do any of that. I used to back in the day. I'm familiar with wet lab stuff, but all of my research now uses the computer programming, data science, things like that. I use these tools and techniques, which are becoming increasingly standard tools across all biology research programs. I use these tools and techniques to study molecular evolution. How do molecules evolve? So more formally, I use biostatistics, data science, and programming to develop and apply probabilistic, AKA using some statistics, models to study how DNA sequences or the genomes evolve, how these things evolve. So for example of that, what I'm showing you down here in this image on the bottom right, this is actually a picture of a protein, right? So we have proteins that they're encoded in genes, and they, we transcribe to mRNA, and then we translate to proteins. And proteins are little useful molecular machines, but they're not static over time. In different species, they evolve to look different, behave differently. And this is an image right now actually of an enzyme. It looks a little different from maybe enzymes that you've seen before. It's a special type of little 3D picture here. And these little balls in the middle represent the catalytic residue. So where does this enzyme bind to ligands to do its thing? All the different colors that you see throughout it, those are kind of visual representations of the amino acids that make up this protein. And they're colored differently in this low to high scale. Different proteins evolve, they undergo mutations and they change over time. And some of these mutations stick around and lead to longer term changes that we see in different species. And we can see some parts of the protein evolve really, really quickly. Those are the parts that are yellower and white in there, and some parts evolve much more slowly, the brown parts in there. And I'm really interested in studying these types of things. How do proteins evolve? And so I developed models to help me figure out things like this, studying different evolutionary rates in proteins. An example of this is, I'm going to talk about hemoglobin. So this is a 3D picture. This is a structure of hemoglobin, the protein that is in your blood, and it carries oxygen around your bodies. Okay. So we have a hemoglobin gene, but so do a lot of other vertebrates. For example, what I'm showing you here, again, here's that hemoglobin up there. This is a little stick figure of a human. This here is a cow, and this here is a fish, tilapia. And what I'm showing you below are a bunch of letters in different colors. These letters represent amino acids. So in fact, there's 20 amino acids that make up our genetic code. And we can represent each one of those with a single letter. So for example, M is methionine, V is valine, L is leucine, and I'm gonna stop there because I'm gonna start forgetting what they are. But each row that I'm showing you here represents a little snippet of the protein sequence, the amino acids that make up the hemoglobin protein. The top row represents human, the middle row represents cattle, and the bottom row represents tilapia. We see a lot of similarities and differences. Over here, we see they all have an M, they all have an L, they all have a W, G, and K, for example, right here. But in other places, they're different. Those represent mutations that occurred in the evolutionary past that led to different forms of this protein. It still functions as hemoglobin. It still folds up into this structure in all of these creatures and carries blood around, but the mutations led the protein sequence to look different. And I'm really interested in understanding the process of how that happens. We also use those statistical models. We analyze these evolutionary changes to, yes, I'm interested in studying evolutionary rates in proteins. That's something that's really interesting to me. But I'm also interested in things like building phylogenies. So evolutionary trees, which you may remember from back in the day in your introduction to evolution class, we actually create these phylogenies. We study species relationships using statistical models to analyze DNA or protein sequences that look a lot like this. This is something called a multiple sequence alignment. That's the most I'll do for vocabulary for you today. But we study these multiple sequence alignments in order to derive different evolutionary insights into life and how life evolves into all of its different forms. A particular interest of mine is to look at how these statistical models, how they actually behave. How well are they doing? How good are the models that we're using? Do they behave well? Do they behave poorly? Do they give us meaningful information about evolution? When do they not? Just because a model is on the computer doesn't mean it's correct or doing it right. Computers can get things wrong too. So we have to study that, what goes on under the hood, to make sure we can actually get meaningful information about biology from these mathematical models. I'm also really interested in different approaches that we can use to identify a particular modeling framework to answer a particular question that we might have. So this is good time to say, what I'm showing you here is an image that looks really similar to what we saw before, right? Those letters on top of each other. I wanted to point out that these methods that I use, these models of evolutionary change, they're the exact same type of thing that we use to study viruses like COVID, SARS-CoV-2, the pandemic that we're currently in, but hopefully making our way out of in the near future. So the methods that I use and the things that I study, they're really broadly applicable. We can think of them just, I'm curious about the relationship among different species. I want to know what protein evolution looks like, or it can be much more applied. I want to track the evolution of COVID across the world. I want to use phylogenies to determine how it moves across the globe and when it infects different people and trace its origins with phylogenies. A lot of really powerful stuff that we can do. So I'll just keep this slide up for a minute while I talk about what students might do in my lab and how in my lab. Here's my lab website. It's https://Spielman, S-P-I-E-L, spielmanlab.github.io. We'll give you some information there. There's my email, and you can also find me on Twitter. Fun fact, a lot of academics, especially in my field, are extremely active on Twitter. It's where we do a lot of scientific networking. So students who join my lab, they often work on these sorts of projects having to do with studying protein evolution, things like that. They also sometimes work on other projects that are more suited to their particular interests, but all of it is going to involve computational work. So there is an expectation that you have some computational background before joining the lab, and we'll be able to discuss that a little bit more in the Q&A that comes up next. I'll give you an example of a couple projects I have going on right now with students. I have one student who's—
Stephanie, we have 1 minute left.
Okay, I have one student who's developing a software in order to make, this actually works well, to programmatically and automatically make images, make figures of multiple sequence alignments. I have another student who's working on, who's creating another software package to be able to facilitate classroom data analysis. And I have other students who are working more on the evolutionary biology aspects here. And cool, that'll be my time. Thanks, everybody.
Thank you, Dr. Spielman. Just real quick for those of you who came in a little bit later, if you have any questions for any of these panelists, you are welcome to write them into the chat and we can ask them during the Q&A. Please make sure that you put the name of the panelist that you are asking the question to at the beginning of your question. So our second panelist is Dr. Mike Grove. If you could share your screen.
Hello, everybody. As Dr. Thomas said, I'm Mike Grove. This work that I'll be talking about kind of briefly really has been done in conjunction with Dr. Courtney Richmond, Dr. Nathan Rule, and I think that's going to be the status of this going forward as well, that this will be a collaborative effort, but in many ways I'll be taking the lead on some of these projects, especially next summer and beyond. So it seemed to make sense for me to talk about them. What we're looking at are zooplankton, and these are probably the least thought about part of aquatic food chains. Everybody knows fish, and you think about at the top of a food chain in the lake, these piscivorous fish eating other fish, and at the bottom of the food chain, okay, I've got some plants there someplace right in the food chain. I've got algae. Oh yeah, I've heard of algae. Zooplankton are that kind of intermediate level. These are herbivores that will feed on the phytoplankton, but then will in turn be eaten by fish. And so they're kind of these intermediate levels in these lake and aquatic food chains. So they're very important because they're connecting the primary production at the base of the food chain to the top of the food chain, the vertebrate predators and the things that we often care about when we go to these lakes and fish. And those zooplankton then are going to be affected by both of these. Keep in mind, these are very small organisms, typically less than 1 millimeter in length. So they're not something that you're going to necessarily see with the naked eye if you're looking in the water. We're looking especially at plankton and zooplankton in reservoirs. And what we mean by a reservoir is simply a lake that has been created artificially by damming a stream or river or something like that. Reservoirs are very interesting and very different in many ways than natural lakes. An artificial reservoir tends to have very large expanses of shallow water and deep water, and you'll have this kind of transition from where the stream or river flows into the lake to the dam, and it will get kind of progressively deeper as you approach the dam, and the habitat will change. So you have these very shallow habitats kind of at the head of the lake where the stream's coming in, and then as I move down towards the dam, it gets deeper and deeper, and sometimes the water will stratify in terms of temperature. We'll have these very specific temperature layers. But I'm really making a transition from kind of one type of habitat, this stream or flowing habitat, into something that's very still water, what we call, a lentic habitat or lake habitat. And so we've got this gradient along the length of the lake here. And we would expect then that the ecosystem function is going to change as I change along that gradient from the head of the lake to the dam. In New Jersey, the reservoirs are different yet again than many places. If I go to, I don't know, out west, and I go to the, Grand Coulee Dam or places like this, you think of with these really super deep lakes. That's not New Jersey, obviously, and especially not southern New Jersey. The reservoirs here are very shallow, typically 6 to 8 feet in maximum depth right behind the dam. The dam, you can kind of think of it as being pushed upstream right into these more shallow areas. And this lake or reservoir is probably going to work differently than something that's much deeper. That's not going to stratify as much, and there are going to be all sorts of other differences that we would think in terms of how the lake is going to function. That difference then is part of what we're interested in, right? How do deep water lakes differ in terms of their ecology from shallow water lakes? We would expect that there's a different pattern in terms of the physical structure of them, so we might expect different ecosystem function. And we don't really have nearly as good a handle kind of globally, I think, in terms of how shallow reservoirs and their ecology works as nearly as much as we do for deep water reservoirs, even the ones that are artificially created. So we'd expect again that there are some differences that we're trying to pull out here. In New Jersey as well, it's very interesting, especially in southern New Jersey, because these reservoirs are often connected in series. So if I look kind of here close to home, right, Elmer Lake flows into what am I looking at here? Palatine Lake, into Parvin Lake, into Rainbow Lake. They're in series. And so if I build up a big population of zooplankton in the lake, they're going to get then exported downstream, potentially all the way to the next lake, then again into a stream. And so I've got this kind of change back and forth down the length of the series from one type of habitat to the other. I'm in a lake, then I'm in a stream habitat. Now I'm in a lake habitat. Now I'm in a stream habitat. And so how does this affect things like zooplankton export? How far downstream from the lake do the zooplankton make it before the fish really consume them all? The zooplankton are not going to build up big populations in a stream because the water is flowing and they can't maintain their position against the current. So in a stream habitat, you're going to have smaller populations, but these lakes now in series are going to act as essentially these big incubators to build up populations that can get then exported downstream. So one of the questions I won't really we don't have time to talk about what we found already, but in terms of how you might be involved, what is coming next, hopefully, and by next, maybe next summer, I'm hoping. One of the questions we're asking is how far downstream do these elevated numbers of zooplankton persist, right? As they export, as I sweep them out of the lake into the stream, how far downstream can I see elevated numbers of zooplankton, or do the fish and other predators consume them very quickly? And as a sub-question here, does this really differ from day to night? And we might expect that it would if you think about the way these work. Most of these dams, actually all of these dams in southern New Jersey, are spillover dams. So the zooplankton are getting kind of swept over the dam from the top of the water column here, right at the surface of the lake, which wouldn't necessarily matter except for the fact that zooplankton will migrate vertically during the course of the day. So during the day, they're down deep to hide from predators. It's kind of been the long-term hypothesis. And then at night, they will come to the surface and feed. So what you might expect in a shallow reservoir here in New Jersey, during the day, the zooplankton are down deep and are not getting swept over the dam. Once night falls, the zooplankton come to the surface, they migrate to the surface, and they may very well be getting swept over and exported in much greater numbers. So we really want to get a handle on how that's happening, or to what extent it's happening. The other thing I might mention very quickly is that's interesting about many New Jersey reservoirs is that they are subject in some cases to these harmful cyanobacterial blooms, these harmful algal blooms or CABs. If you see this map from the state of New Jersey DEP from 2019, the green dots are lakes in which these harmful algal blooms were reported. Here's a picture of one in Elmer Lake, one of the lakes we worked at in that year. But these lakes here in Cumberland County, right, are all in series. This is on the Salem River watershed here, all in sequence. And so one of the other questions we're really interested in is, do these cyanobacterial blooms, when they occur, does it change the community of zooplankton that we find in those lakes? And so what our thought would be to go out and sample these lakes that are in series here on the Salem River. We've got at least 5 lakes where these blooms have been known to occur on occasion. So if we can kind of sample them before, during, and after a bloom, we hope to be able to kind of get a handle on just how much those harmful algal blooms are affecting the zooplankton, and then ultimately potentially the fish populations because of that. So how can you get involved? What would you do if you're involved in our lab? A lot of it is field work. So on a weekly basis, we go out and collect zooplankton from the reservoirs. You would bring them back into the lab to identify and count. So that's a lot of the work is microscope work, kind of IDing and counting the zooplankton.
Dr. Grove, you have 60 seconds left.
Yep, okay, great. We've had students that have been very involved in the data analysis and manuscript writing. We have one paper that's already been published with undergraduate authors. There's another coming out hopefully here in the next 6 months. We're hoping to get it out and published. So that's certainly something you can do. And lots of our students present at a whole variety of conferences, scientific meetings, local level. So here's a picture of 2 of our students. This is from one of the summer symposia. We've had students go to national and even international meetings. During the COVID, 2 of our students presented virtually in an ecology conference in Finland. So that was, I think, fun for them and fun for us as well. So anyways, just a kind of quick overview of what we do. So happy to take questions later.
Thank you very much, Dr. Grove. Our third panelist is Dr. Claude Krummenacher. If you could share your screen, please, and remember to unmute yourself.
All right, can you see my screen?
You mean your picture of yourself?
It's not my picture. It's a picture I got from the web. So we in my lab we study basically viruses, and the model virus that I've been working on is herpes simplex virus. And from the studies of the initial interactions between herpes viruses and their host, we actually derived new projects that are more cell biology oriented or even cancer biology oriented. So this is what I'm going to present to you today. You probably all have heard about herpes. Hopefully you don't suffer from it, but these are very prevalent viruses, and many people are actually infected with herpes simplex virus type 1, which is the virus that typically causes oral herpes, and the main symptom being a cold sore when the virus reactivates from being latent in some neurons at the center of your head. HSV2 is the genital form of the virus that causes the sexually transmitted herpes, although HSV1 is now also a prevalent virus that causes genital herpes. There are no vaccines against HSV, And there are some drugs that alleviate the symptoms, but they don't prevent infection. So in my lab, we are interested in the initial interaction between the virus and the cell. You know that all viruses need to infect a cell in order to replicate. So we are looking at how HSV, herpes simplex virus, gets into a cell. So the first question that we ask is, how does the virus interact with the cell in order to get in and start infection? One of the projects that is ongoing in the lab is trying to design drugs that can actually block important interactions that the virus needs in order to get into cells. We are also interested in the immunology against herpes simplex virus, especially the role of cells from your immune system called natural killer cells. These are cells that go around and find virally infected cells and kill them in order to prevent virus propagation. Now, these natural killer cells are also very important in fighting cancer cells. So we have also a project that look at cancer cells. So I will detail these projects now. Everything started by looking at these molecular interactions between the virus proteins shown here on top in these very colorful models and cell receptors. These are specific proteins at the surface of the cell that the virus binds to and allows the virus to get in. Now, the molecule that we study, the protein we study, is called nectin-1. And the virus will bind to nectin-1 in order to get in. Now, this interaction is critical for infection. So what we are trying to do is to design drugs that will bind to the virus and prevent the virus to bind to nectin-1, and therefore we would block infection. Now, this is a collaboration with Dr. Chun Wu from MCB, and he is a specialist of studying protein structures. We found little drugs, and then we are testing them in my lab using a functional assay called a plaque assay. So on the right here, you see all these little light spots on that purple background. Every spot is the beginning of an infection. So we can quantify infection and we can quantify the effect of these drugs. So my students do these assays. They can do infection. They can block virus infection using these drugs and testing these drugs. Now, nectin-1, which is the receptor that the virus uses, is not there at the cell surface just to help viruses to infect us. They have another role. And the role of nectin-1 has been quite difficult to assess. It's involved in cell adhesion. It allows cells to bind to each other and attach to each other. But it has a role now that we just found in immunology, in immunity. And it helps these natural killer cells to bind to their targets. And you see on this cartoon on the right, an electron microscopy image of 3 natural killer cells latching on a cell that they are trying to kill, right? And nectin-1 is involved in this interaction between natural killer cells and the target cell. And we first thought that the virus was affecting the function of nectin-1 so that it would avoid NK cells to come and kill herpes virus infected target cells. So that's what we are trying to study. And that's our immunology project. Now, as I mentioned, these NK cells are also involved in killing cancer cells. So we were interested to find out whether nectin-1 actually plays a role in NK cells killing cancer cells. So the role of nectin-1 in cancer development is not very well known. So we are doing that right now using breast cancer cell lines in the lab, trying to figure out whether nectin-1 is expressed on these cells. And the next step of the project will be to look at whether the presence or absence of nectin-1 on cancer cells affect their susceptibility to be recognized and killed by the NK cells. The ultimate goal will be for us to try to help these NK cells to actually kill more effectively cancer cells. So we are focusing on this protein, Nectin-1. We started by studying its role as a receptor for herpes simplex virus. And now we're looking at its role in the immune response and more specifically, the immune response against cancer cells. So these last two projects do not directly involve viruses, but all these projects have a common denominator, which is this molecule Nectin-1, which we also associate with all the cousins in the same family, Nectin-2, Nectin-3, Nectin-4, and so on. So we look at the panel of this Nectin family and their roles in these three aspects of functions: immunology, cancer biology, and virology. So there's a
Dr.
lot.
Krummenacher, you have 1 minute left.
All right. So we do a lot of cell biology in the lab, and my students have to learn how to culture mammalian cells and human cell lines, and how to work properly in a sterile environment in order to grow these cells and do various assays in microscopy, in viral infection, and so on. So these are the current people in the lab. And when we don't work, we eat herpes pizza and drink herpes beer. No, this is not quite true. We work all the time. I want to thank you. And if you have questions on, about all this, you can ask in the chat or at the end during the Q&A. Thank you.
Thank you very much, Dr. Krummenacher. Our fourth panelist is Dr. Svjetlana Kruse. If you could share your screen and unmute yourself.
Okay, can you see my presentation?
Yep, it's good.
Okay, so my name is Dr. Kruse, formerly Vojvodic. I'm here associate professor in biology department, and I'll give you a little overview of what's going on in a social insect lab. So before we go into social insects, one of the big components of my lab is gut microbiomes. And so those are all the bacteria that are found inside the gut. And humans, bees, all the animals really have these microbial communities in their intestines that have been now linked to a lot of different things. In humans, we know that they can have impact on immune responses, absorption of nutrients, and other things. But more recently, what we have found out is that some of these microbes have been linked with diseases, and specifically any changes of the microbiome. So abundance of one microbe compared to a healthy individual, or disappearance and appearance of different microbial communities. So anything from allergies, celiac disease, even gastric cancer have been linked to some of these changes, and we call that dyssymbiosis. We have also observed that individuals that have specific behaviors like autism have a slightly different microbial community, what has been found in what you would normally call a healthy individuals. So it seems that there is some kind of component between the gut microbiome and behavior. And my lab is interested in sort of tackling some of these issues using honeybee as a model to address how do gut microbiome influence behavior, specifically individual behavior, social interactions, and what are the mechanisms involved in these things. So what are social insects? Just a little bit of a background information. So those are honeybees, bees, wasps, all the ants, as well as termites. And those are highly social animals or insects. Besides humans, they really have the most complex sort of interactions. And we can assume that some of these interactions also lead to exchange of microbial communities. And we have seen and observed some of these behaviors, as well as some of these microbes being transmitted. When it comes to honeybees, here we have on this picture worker bees. The one with the dot here is a queen. You can see that she's much bigger. And these bees stand on a frame that has this worm-like things. Those are honeybee larvae. And then once the larva kind of grows to a different a size, specific size, they're going to be capped and they undergo pupation, and then the baby bee comes out. Now, the honeybees, like humans, acquire microbiomes twice throughout their lifetime, so they actually have to acquire it after they're born. Like human babies. And so, and the way they acquire it is through food exchange. They also acquire it during the larval stage. So during the larvae, they acquire all these microbes, then during metamorphosis, they lose it, and then they have to acquire it again as a young adult bee. So we are interested in several different sort of these time points during the development. When do bees acquire, what do they acquire these microbes, and how do these microbes influence bee behavior? So this is just an overview of what we are interested in my lab. So we have what is known as a microbiome gut-brain axis, and that is that there is a direct link between the brain of the bee as well as the microbiome or the bacteria found in the gut. And this is a two-way street. So the microbes don't go into the brain, they stay in the gut, but they do send signals through chemicals that can change and alter bee behavior. So what we tackle here in my lab is how any changes in the gut microbiome that we actually induce ourselves influence individual bee behavior. So we do the associative learning of bees, and we also look at social interactions. How do bees interact with each other, and does that change as their microbiome changes? And the third aspect of this research is looking at how do these changes, can these changes be reflected in the brain genome as well as the gut tissue genome? So we are doing RNA sequencing to address some of these other questions, which is really the mechanisms behind it. So this proboscis extension reflex is really just training the bees to associate a specific odor with a reward. Bees are very smart. This works really well. And so here we have a bee that's been strapped in a harness. You can give her a toothpick with sugar, touch her antennae, she will extend her tongue, and you can associate that with a specific smell. And after a few times, she will actually start to extend her tongue out as soon as she smells it because she expects the reward. So it's like a Pavlovian test that you guys probably studied in psychology. So we do this with honeybees. We also can tag bees and develop social networks. So we can analyze the videos of bees exchanging food like here, or grooming. We can also look at the proximity where individuals are within the social network. So you cannot really study social networks, except in humans, which is kind of hard. So that's why we actually use bees. Furthermore, what we do is we isolate a lot of gut microbial communities, so individual bacteria. We have quite a lot of those cultured on plates. Here we have Sarah culturing some of these microbes. And then what we do is we take those baby bees and we actually keep them in the lab, and we introduce these bacterial communities to them. So we can actually regulate what microbes are present at what time. And then we can use these bees to address some of these individual as well as group level behaviors. So there's a lot of sort of microbiological work being done in the lab right now.
Dr. Kruse, you have a minute left.
Okay, so here we have a bunch of students from this summer working and analyzing bacterial communities. We also do quite a bit of beekeeping. We have an apiary or a bee yard here on campus where we keep our bees, and then we can actually use them for research. We have also established a new club. It's a student-run club, beekeeping club, and hopefully we'll have more students involved with this. And what else? Also, we have ants in the lab. So we collected ants early in the spring. We can color those ants and also work on their social networks and disease resistance. My students also present a lot at the conferences. We have some regional conferences like the ones that take place at Rowan, but I have also taken students to international conferences, Entomological Society of America conference, where they did talks or poster presentation. Here we have a current lab picture as well as all the former students. And if anyone is interested in working with me, just please send me an email. If you have a resume, maybe attach that. Let me know what you're interested in. Thank you.
Thank you, Dr. Kruse. Our fifth panelist is Dr. Patrick Crumrine. He is teaching a class right now, so he made a video for us, and let me upload that for you now.
Hello, biology students. My name is Dr. Patrick Crumrine. I apologize for not being in person today, but I teach a class at this time, so I made this video to tell you a little bit about the work that I do here at Rowan. So I've been here at Rowan for about 15 years, and my research is in the area of aquatic community ecology. And I'll talk a little bit about what that means. And the aquatic part represents freshwater ecosystems, and the ecosystem that I spend most of my time working in is the temporary fishless pond or wetland. And these are really great areas for aquatic insects to live because they often lack fish predators. So sometimes these ecosystems dry up in very dry years, and in other years that receive more rainfall, they stay full of water. So they're an excellent place to do community ecology. So the community part describes the interactions that occur between species. And I'm interested in how predation interactions play out in this particular ecosystem. So these three insects are three of the species that I've worked with quite a bit over the years. On the left is the top predator in the communities that I work in. This is a diticid water beetle larva in the genus Cybister. This insect on the right is the common green darner dragonfly, and this is the aquatic nymph stage. And on the bottom is the blue dasher dragonfly. This is also the aquatic nymph stage. So the interaction that's depicted here is called intragild predation. So the arrows represent predator-prey interaction. So the diticid can eat the anax. The diticid also eats the pachydiplax, and the anax can eat pachydiplax. So these two predators are engaged in a predator-prey interaction themselves, but they also compete for shared prey. So intragild predation is one of those community level interactions that I study. And ultimately, what we try and do in ecology is better understand patterns of distribution and abundance of species in the biosphere. So that's represented here in this distribution map of Anax junius, which is very widely distributed throughout North America, as well as the patterns of abundance, which is depicted in this panel of figures on the right that shows the abundance of Anax in ponds throughout its distribution at different times of the year. So I'll tell you a little bit more about the work that I do that's focused on the role of size structure and how that influences the outcome of predator-prey interactions. So Anax junius, the common green darner, is one of the species I've spent a good deal of time studying. It goes through these huge changes in body size as it grows and develops. So these are instars, different developmental stages of this aquatic insect. And you can see that in the 7th instar, this individual is quite small, But in the final instar, the insect is much, much larger. And these are all the same species, but just different developmental stages. So this is what we describe as a size structured population. So lots of different size individuals, the same species in the population. So size structure has a big impact on the outcome of predator-prey interactions. So I study how interactions between individuals, like cannibalism, affects the survival of prey organisms, and then how interactions between different size structured predators affect the outcome of those predator-prey interactions, and ultimately community structure, or the types of species that we find in communities and how many of each of those there are. The way we do this is through replicated mesocosm experiments. So these plastic tubs are basically like mini aquatic ecosystems, and we'll put different combinations of different species and different size classes in these mesocosms and do predation trials and see which ones survive, what kinds of behaviors and things might influence their survivorship. And again, that's to ultimately better understand patterns that we see out in nature in whole large ecosystems. So size structure is one factor that influences predator-prey interactions that I work on. And I'm also interested in things about the physical habitat and how that affects the outcome of these predator-prey interactions. So I've done some work that looks at how structural habitat complexity affects predator-prey interactions in that same group of insects. And the pictures that you see here are on the left. This is a plant called mermaid weed, and it has different growth forms. So there are some stems that have many, many, many leaves, and there are other stems that have fewer leaves and are less complex. So from an aquatic insect's perspective, this is a very, very different kind of habitat and can influence interactions between individuals and between species. So on the right here are mesocosms that have algae growing in them, and algae is also something that varies in nature. Some ecosystems have lots of algae suspended in the water column and on attached surfaces, while others don't. So a student who worked with me this summer looked at the interaction between habitat complexity and algal abundance and how that affected the outcome of these predator-prey interactions. So most of this work that I've described with aquatic insects is work that I do at University of Virginia's Blandy Experimental Farm. So during the summer, I relocate down to Virginia and coordinate an undergraduate research program funded by the National Science Foundation. We've had Rowan students participate in this program in past years, and I would be eager to tell you lots more about it if you're interested in applying for that. So the work that I do here at Rowan is focused a little bit more on aquatic turtles and their ecology and how urbanization and land use change affect population structure. So the image that you see here, this aerial shot, shows Abbott's Pond and the stormwater retention pond that's next to Holly Point Commons. And these are both habitats that aquatic turtles use, particularly eastern painted turtles. So the students pictured here are students that worked with me about 5 years ago, and that's Alyssa Ambacher and Kelsey Wright. Matthew Gladfelter worked with me in 2017, and he is now a PhD student at the University of Auburn working on harmful algal blooms. So he got a start here at Rowan working on turtles, and then he shifted to looking at algae. So the work that we do with aquatic turtles, like I said, is focused on population structure. So we're interested in how urbanization affects how many males versus females there are in a population, and also the age structure of that population. So in urban environments, we might expect there to be greater risk for turtles that leave ponds to find suitable nesting habitat, and that's females. So females are hypothesized to be more at risk of being perhaps run over by cars, picked up by people, or otherwise harmed when they leave the pond. Whereas males don't have as much need to leave a pond because they don't have to go find suitable nesting habitat outside of the pond. So I'll show you one result slide here that was generated by work that Matthew Gladfelter carried out. He looked at ponds across a gradient of time since urbanization, and that's here on the x-axis, and on the y-axis is proportion female. So he was able to show that landscapes with a long history of development or urbanization tended to have fewer females in the population. So this is some evidence to suggest that if there's a longer signature, a longer time since urbanization, there are fewer females, which supports the hypothesis that urbanization leads to greater abundance of males in populations because females are leaving ponds and perhaps being run over by cars when they're trying to find suitable nesting habitat in more urban areas. So if any of this seems interesting to you, or if you want to learn more about aquatic turtle population ecology or aquatic insects and predator-prey interactions, or if you want to learn more about the REU program that I help coordinate at University of Virginia, my contact information is here, and I'd be happy to talk with you more about it. Thanks for your time.
Okay, so now is the time for the Q&A, and we encourage you to post your questions in the chat. We're going to be addressing a number of the questions that are in the chat first. And then if there's time at the end, we may open it up to asking directly via your mic if you are interested in doing it that way. Please note that Dr. Richmond is also here, as Dr. Grove and Dr. Richmond share a research program. So our first question is for all of the panelists. And that question is, could you please explain how students can apply to work in your lab? And I'm just going to move through the panel, so I'll ask that to Stephanie first. Or Dr. Spielman,
yeah,
sorry.
Stephanie's fine. Yeah, let me say, this is the only place I've ever worked where it is the culture to say Dr. So-and-so instead of the culture to refer to their professors by first name. So that's where I'm coming from. You can all call me Stephanie if you're in my lab, please. I believe a message was put into the chat. I have a link in there where you can read about how you can go about joining my lab or reaching out to me about how that works. I generally recruit students out of one of my classes. That's either the data science for biologists class, or I'm launching a new class coming up this spring, which I'm really excited about, called Genome Sciences. And it's a very hands-on class where we're going to really learn how to work with genomes, what's in a genome, how do genomes evolve, how can I interpret them, how can I use them, things like that. So yeah, taking one of my classes is going to be your entryway into joining my lab, because my lab's a little bit different from others. It's a very different topic from a lot of the other research programs dealing with molecular evolution. And also we use different sorts of skills like computer programming and data science skills, which you do need to have a bit of a handle on before coming in. And of course, we will expand your skills tremendously in that domain once you're in the lab. I will also take this moment to say this is my 4th year at Rowan University, so I'm a senior over here. I've had a relatively smaller number of students over the years because I've had fewer years here. But the students who come through my lab get jobs. They leave here and they get jobs because the types of skills that I teach you are the type that you learn in my lab and also in my classes are types of skills that are not generally taught, but they're assumed you're just going to go out and learn them on your own. That's really challenging. So my lab does really provide you with a lot of professional development if you're interested in anything in the biotech sector broadly interpreted. Cool.
Great. Thank you. Dr. Grove or Dr. Richmond?
I think the way we've typically done this in the past, we will send out an announcement through all of the bio majors sometime probably early spring, January or so, once we have a handle on who's coming back with us for the following field season. Typically what we'll just do is ask everybody to send us a brief application, maybe a copy of your CV or resume, and ask you to give us a bit of background information. And then, to be honest, we've usually done an interview process. We've had enough interest that we've had, unfortunately, have not always been able to take everybody that's interested. We've been able to take the majority of the students that have applied, but we will kind of go through an interview process and kind of See if, I mean, is this the right fit? I mean, for what you want to do, at least now, even if it's not your long-term career aspirations to work in ecology. So.
Thank you. Dr. Krummenacher?
Yes, so in my lab, most of the students that are working in the lab now are actually students who just came to my office or emailed me and asked to come to the lab. If you're really motivated to do this type of research, you're going to look around at who is available to do research in cell biology or any of the field that you've heard today. Do your homework, look at what these people are doing, and then contact me, send me an email, come to my office, send me your CV. And what I consider very important is that you have to demonstrate that you are very motivated to do research. The research that we do is in my lab is complicated. Things don't always work, so it can be frustrating at times. When it works, it's very rewarding, but doing research is something that you won't really enjoy if you're not motivated to do it. So for me, motivation is very important. So if you really want to do it, contact me, contact the people that you are interested in working with. That is one of the aspects. The other aspect is, as Dr. Grove mentioned, the program. There's a summer research program for which there are positions open. These are funded by stipends. And in the spring, there may be positions opening in my lab at this point. And then at that case, if I have several candidates who want to join the lab, then there will be some interviews and the selection. But I must say that it depends on the timing. So ask for joining labs in the spring, early in the spring for the summer, or at the end of the spring for the fall, or things like that. Right now, I receive still emails from students who want to join now, or even for the spring, and it's already a bit too late at this point. So get your timing right. Don't wait. Just ask. And if I cannot make a decision right now, I will tell the students, contact me again in December. I will know better about how the spring is going to be. But I would say I don't mind being bugged by students who want to join the lab. And I think it shows motivation, it shows interest. But I must say that I keep my lab small because of the type of research we do. We cannot have a lot of people working at the same time in cell culture hoods or with viruses around or things like that. So I keep my lab small, and I must say that I say no to a lot of students who want to join my lab. So it is competitive. It's unfortunate for all the students involved who want to be involved in research that maybe there are not enough positions. But again, if you show motivation, if you show interest, then you have more chance to join a lab like mine.
Thank you, Dr. Krummenacher. Dr. Kruse?
Yeah, so generally for my lab, I have done both advertised positions, usually because of the nature of the research. It's very intensive during summer. Students are required to be here over summer. I also ask often students to, if they can join multiple semesters, because it takes a long time to train you guys. And then if you're gonna just leave after one semester, that's a huge investment of my time. So I prefer to have students who are, available for several semesters to work in the lab. So again, in the past, I have advertised for positions primarily for the summer. I have also, received emails before from students who are highly motivated, or even, students who have talked to their other professors, and they would come to me and say, hey, I have a really great student, highly motivated student that's looking for the lab. Would you be willing to take this person. And so I would interview them and things like that. So I would say definitely do your homework, as Dr. Krummenacher said. Look at the website, the labs of all of us. We have a lot of information there. If you know any of the students that are currently working in my lab, definitely reach out to them and ask them, how is it to work with me? What are we doing? Things like that are pretty important. So highly motivated students, timing is pretty crucial. So if you're already senior, most likely I'm not going to take you because you're going to be gone in a semester. I kind of hate to say that, but it is true that it is my time also that I'm investing in training you guys. So I'm also trying to get something out of that by having you actually a little bit longer in the lab. But I think once you get in, obviously being serious about it, taking things seriously, committing to actually doing the work. There are rewards, which is traveling to different conferences, having access to me to ask me questions anytime you want, lab meetings, learning primary literature, broadly, training in behavior, genomics, microbiology. So there are definitely benefits, but it is competitive. So right now the lab is full. We have 8 people in the lab, which is kind of the limit I can handle at the moment, but it doesn't mean it's not going to change before the summer. So if you are interested, talk to me about it. You can also email me and
Thank you. This next question, some of you have already addressed this in your last comments, but if you haven't addressed this yet, the question is, what year should students get involved in their research? And do you accept first years? And kind of combined with that, the question is, do students need experience before applying to work with you? So if you haven't addressed that yet, if you could, we're going to go random, whoever would like to respond to that one. Go ahead, Claude.
Maybe I'll start with that. As Dr. Kruse mentioned, I also want students who can commit to several semesters in the lab. If you come to me in September and said, I'm graduating next May, can I join your lab? It's very unlikely that I will say yes. Unless there are exceptional circumstances, and I've taken students starting their senior year, usually you should not wait until that time to do research. I like students to have a little bit of experience. So if you're biology students, I usually like students who have Bio 3, which is Intro to Cell Biology. But I can take students that have done Bio 2, the Intro to Genetics. Just having a little bit of lab experience, knowing how to pipette, having an idea of how to calculate dilutions and things like that is definitely a plus when you join the lab. So I would say for me, I'll take sophomores probably the most, and maybe starting juniors. But don't wait until you're senior to ask to join the lab.
Thank you. Did anyone else want to respond to that?
I'll just again direct you to the website that was posted in the chat for me. I'll take anybody early, but it really is depend on some skill background that you have. Notably, my data science for biologist course is open to sophomores, but it's a bit tricky to get into. So it will be offered again next spring along with the genome sciences class. So if you think that doing research with me is something potentially interesting to you and you're not graduating this spring, you might want to look into registering for one of those or reach out to me to discuss how might you go about registering for classes in the future for these opportunities.
Great. Go ahead, Dr. Richmond.
I was just going to say for us too, to sort of back up what we're already hearing from Dr. Krummenacher and Dr. Spielman. I don't think that there's too early, or maybe even more so than what they're saying. I don't think that there's too early for us. I think we can really train our students for what folks do with us. And I think that's true for a lot of people, that like having students earlier and being able to have them longer term, which we have just heard here, is a better thing. So I think, to the students who are listening, if you're thinking about getting involved in research, it's definitely not too early to start to talk to faculty and find who might be able to take you, and then they can tell you themselves what they might want you to have before you could start working with them, and you could work that out with them.
Thank you. I think we have time for one last question. And the student asks, if none of the research really goes with what they want to do professionally, do you still recommend that students do research with you, even though that doesn't align perfectly with what they want? Maybe one of you could address that.
I think it's
Yes.
absolutely, appropriate for students to do something quite different than what they, at least now, think their career interests are. Some of the most motivated and best students I've ever had work with us knew, the very best student, I would say, knew all along he wanted to be an osteopath. His family were all osteopaths. He had no interest in becoming an aquatic ecologist. I think he enjoyed it. I think he learned a lot from it, and I think it was a great relationship. I think absolutely it's often a good thing to do something very different just to try it out. I mean, if nothing else.
Yeah, I would agree with that. I see everybody else on the panel nodding and everything. Yeah, I would say that everybody should try to get almost everybody should try to get a research experience as an undergraduate. And if you can't get one here at Rowan with our panelists here or any of the other professors, you can go outside of the biology department to get a research experience. And you can also get research experiences off campus. And along with that, let me just note again that if you go to the biological department calendar, you'll see that there's a number of workshops that are coming up that include things like how to find the correct, like a graduate school that you're interested in, how to apply for that, how to develop a good CV or resume, how to develop a good cover letter, and other kinds of workshops. So those, watch for flyers and emails that will be around the department, and then also watch on the biological sciences department calendar for those links. All right, we appreciate so much everybody attending today, and thank you very much to our panelists. So thank you very much to everyone. Please remember to watch for the upcoming seminars, and I'm excited about Dr. Keck coming up next. Thank you very much, and have a good week, rest of your week. Thank you, everyone.
Thanks, everyone.
Thank you all.